Welcome to chapter 16 of Cassette and Dose Toxicology, Toxic Responses of the Nervous System. So this chapter highlights how chemicals and toxins affect brain and nerve functions, leading to various neurological issues. I hope this session enhances your understanding of neurotoxicity. So let's dive into the key mechanisms and effects of neurotoxicity. Before we jump into the details, let's take a quick look at the topics covered in this chapter. So we'll begin with an introduction to neurotoxicity and a foundational overview of the nervous system, covering key elements like the blood-brain barrier, axonal transport, axonal degeneration, neurotransmission, and development of nervous system. So from there, we'll explore how toxicants affect the nervous system function and the mechanisms behind the neurotoxic injuries and the roles of neuronopathies, axonopathies, and myelonopathies. And we'll also understand how environmental and developmental exposure contribute to neurodegenerative diseases, and how the in vitro and modern models, from high throughput analysis to computational models, are revolutionizing this neurotoxicology. And finally, we'll discuss about the epigenetics and neurotoxicology. Introduction to neurotoxicity. So let's understand with the fundamental question, what is neurotoxicity? So neurotoxicity refers to the ability of a toxic substance, whether it is natural or synthetic, to cause harmful effects on the nervous system. So this can impact everything from motor control to cognition and emotional regulation. So now it's important to distinguish between two key terms, that is neurotoxins and neurotoxicants. So neurotoxins are typically natural substances, like those found in snake venom or certain marine organisms. So these neurotoxins are directly disrupt the nerve cell function. For example, botulinum toxin, which is produced by Clostridium botulinum bacteria, which blocks nerve signals to muscles, which causes paralysis. And the next key term is neurotoxicant. This includes a broad range of chemicals, both natural and man-made, that interferes with the nervous system function. So these can affect the brain during the development or in adulthood. So examples: lead, mercury, pesticides, and some of the industrial solvents. So let me tell you a story here. In the mid 20th century, many homes were painted with the lead-based paints in the US. So over time, the paint which is there was chipped and turned into dust. The children living in that area were started like by inhaling the dust and putting their paint chips into their mouths. So this was a serious issue, and children's exposed to high level of lead, uh, which often showed the lower IQ, learning difficulties, hyperactivity, and aggression, and some of them are developed delayed development. Even hearing and speech issues were observed. So later, the U.S. government and regulatory bodies were like banned this lead-based paints in residential buildings in 1987, 1978. Overview of nervous system. The nervous system is a complex network that plays a crucial role in coordinating and regulating bodily functions. And the nervous system is responsible for processing information and controlling movements and facilitating the communications between different parts of the body. So, for example, right now you're sitting in front of the system or laptop, right? So your eyes are focused on the screen and your ears are picking up my voice, and at the same time your brain is processing my every word I say and trying to understand and remember. But at the same time, your heart is beating and your lungs are breathing, your stomach is digesting the breakfast you ate, and the body temperature is being regulated. all without even thinking about it, right? So it's it's all managed by your nervous system, a silent, tireless network that constantly working behind the scenes. So without it, you won't be able to move, think, feel, or even survive. That's how powerful and essential your nervous system is. So the nervous system is broadly divided into two main parts: the central nervous system and the peripheral nervous system. The central nervous system consists of the brain and spinal cord. It serves as a control center for processing information and making decisions. So let's look at the brain. So the brain is again most complex organ in our body, and it is responsible for higher cognitive functions, motivation, motions, memories, and sensory processing. And it is further divided into several regions, mainly cerebrum, cerebellum, and brainstem. The cerebrum, it's the largest part of the brain, responsible for voluntary actions, thought process, and sensory precisions. And the cerebellum, which is located at the back of the brain, it coordinates the movements and balance. And the brain stem, which connects the brain to the spinal cord and controls vital functions such as breathing, heart rate, and blood pressure. And the other part of CNS is spinal cord. Spinal cord extends from the brainstem down the vertebral column. It serves as a channel for signaling signals between the brain and the rest of the body, and is involves in reflex actions. So the other part of the CNS is the other part of nervous system is peripheral nervous system. The peripheral nervous system connects the CNS to the limbs and organs. It is further divided into two main parts, main components. One is somatic nervous system and autonomic nervous system. So somatic nervous system, this system controls voluntary movements and transmit sensory information from the skin, muscle, and joints to the CNS. For example, if you just think that if someone asked you to raise the hand, so you first heard the instruction and your brain processed it and then sent signal through your somatic nervous system to the muscle in your arms and shoulder, and you consciously decide to move. So this is called voluntary movement. So this is what exactly the somatic nervous system controls. And the next one is autonomic nervous system. So this system regulates voluntary bodily function, involuntary bodily functions such as heart rate, digestion, and the respiratory rate. So it is further divided into sympathetic nervous system and parasympathetic nervous system. So imagine that you're walking alone at night and suddenly you heard a loud noise behind you. So instantly what will happen? Your heart rate rises and the breathing uh, comes faster and your pupil dilates, and your body, eventually your body gets ready to either run away or defend itself, right? So that's your sympathetic nervous system, which your body's built-in fight or fight responses. So this prepares your body to deal with the dangers by increasing energies and alertness. And at the same time, you imagine that you realized that it is just a dog knocking over a trash can. And this time you will calm down yourself and your heart rate slows and your breathing turns to normal and your body parts get relaxed and digest again. So this is your parasympathetic nervous system taking over. That means like restoring balance and promoting rest and digest functions. So all of this happens automatically. So without you having to think about it, that's how the autonomic nervous system work. quietly managing your bodily vital functions every second of your life. Now we'll try to understand the neurons and their parts. So neurons are the fundamental building blocks of the nervous system. The neurons are responsible for transmitting information throughout the body. So they play a crucial role in processing and communicating signals. If you see one neuron transmits signals at lightning speed, that is around 400 km per hour, which enables everything from the reflexes to complex thoughts. So these neurons are specialized cells which consist of three main parts: cell body, dendrites, and axon. So the cell body, it is also called as soma, and this is the central part of the nerve, and it contains nucleus, which houses the cell genetic material, and it is responsible for maintaining the cell health and functionality. And surrounding the cell body, you find a branch-like structure that is called dendrites. Uh, these are responsible for receiving signals from other neurons or sensory receptors. So dendrites increases the surface area of the neurons, which allows to form connections with the multiple neurons. And the last part is axon. This axon is a long and slender projection that transmit electric impulses away from the cell body to the other neurons and also muscles and glands. So the axon can vary in length, which can be around 1 meter long in humans. And at the end of the axon, you will have the terminal buttons that are released neurotransmitters into the synapses. So if you see, one neuron can form up to 10,000 synaptic connections, and the gap between neurons facilitate the communication. So many axons are covered with the fatty layer known as myelin sheath, which acts as the insulator. And apart from these neurons, you also find some other cells, neuroglial cells that support these are the supporting cells of the nervous system, which are divided into those found in the peripheral nervous system and central nervous system. If you see the cells, supporting cells of the peripheral nervous system, you have satellite cells and Schwann cells. These satellite cells are surround neuron cell bodies in ganglia and regulate levels of oxygen and carbon dioxide, nutrients, and other and neurotransmitters. Schwann cells, these are primarily known for forming the myelin sheath around axon, which is essential for rapid nerve impulses transmission. So, if you see in Charcot-Marie tooth disease, that is CMT. So in this, it is a genetic disorder. Like this will this is a genetic disorder where the Schwann cells will get affected in this, and resulting the defective myelins and progressive loss of the muscle tissues and touch sensation will happen in this with this disorder. And yeah, we'll see about the central nervous system cells. The supporting cells of central nervous system are ependymal cells. They are epithelial cells, helps produce, circulate, and monitor the CFS, that is cerebrospinal fluid. And other cells are oligodendrocytes, which are responsible for forming the myelin sheath around the axon in CNS. So, which provides the structural support. So, if you see in multiple sclerosis, the immune system attacks the myelin sheath formed by oligodendrocytes. So this leads to the disruption of nerve signal transmission and causing symptoms like muscle weakness, vision problem, and coordination issues. And the other type of cells are astrocytes, which are mostly abundant in glial cells in the brain, and they are star-shaped cells which maintains the blood-brain barrier and regulates ions and nutrient levels. And the microglia cells, these are resident immune cells of the CNS. So they are small and motile cells that act as scavengers, removing cell debris, pathogens, and damaged neurons. For example, in Alzheimer's, microglia become overactive and contribute to chronic inflammation in the brain. So instead of protecting nerves, they may damage them, releasing harmful substances. Uh, let's discuss the blood-brain barrier. So the concept of blood-brain barrier was first introduced by Paul Ehrlich in 1985. The German scientist is renowned for his contributions in the field of microbiology. Especially, he developed chemotherapy and understanding of the immune systems. And Ehrlich was also a pioneer in using dyes to stain tissues for microscopic examinations. He wanted to understand how different dyes interacted with the various organs and tissues. So, in his one of the experiments, dye exclusive experiments, Ehrlich injected dyes into the bloodstream and observed that well, most of the organs were stained, but the brain and spinal cord remained untouched. So this led him to propose that the central nervous system was protected by a unique barrier that prevented certain substances from the entering of this. So although he didn't understand fully the mechanism at the time, but his work laid the foundation steps for the blood-brain barrier concept. So the blood-brain barrier is composed of specialized endothelial cells and tight junctions and supported by astrocytes and transport proteins. The primary functions of blood-brain barrier includes it protects the brain from potential harmful substances such as toxins and pathogens, while it allows essential nutrients to pass through. And also, it helps maintaining the brain microenvironment by regulating ionic concentrations and removing the waste products. So the barrier selectively allows certain molecules such as glucose and amino acids to enter into the brain, while it blocks others, including large and hydrophilic molecules. So that's why the treating brain diseases is so hard and challenging. It's because most of the medicines cannot cross the blood-brain barrier. So, however, the blood-brain barrier is not perfect. Certain regions, like the circumventricular organs, lack the tight junctions, which allows the selective chemical entry. And in newborns, especially premature infants, the blood-brain barrier is not fully developed, making them more vulnerable to neurotoxicants like unconjugated bilirubin and the hexachlorophene. So the integrity of this barrier is crucial for maintaining the brain health. Disruption of this barrier has been implicated in various neurological disorders, which includes multiple sclerosis, Alzheimer's disease, and the strokes and infections. Some pathogens can compromise the blood-brain barrier, leading to the conditions such as meningitis. Yeah, let's understand the energy requirements of neurons and nervous system. The brain, despite representing only about 2% of our body weight, consumes a disproportionately large amount of our total energy, that is roughly 20%. Why? Because neurons are constantly working, maintaining neuronal functions, transmitting signals, and supporting the complex processes, and coordinating everything from your heartbeat to your thoughts. Both the neurons and cardiac myocytes generate and transmit electric signals. The neurons are for communication and the cardiac cells for heartbeat coordination. So this function demands a high metabolic rate, primarily to maintain and restore ion gradient across membranes. And this both the cells types rely heavily on aerobic respiration to meet these energy requirements. And the neurons are post-mitotic. That means they do not divide. This makes energy homeostasis essential for their survival and function. Even at rest, neurons must produce large amount of high energy phosphates like ATP to support frequent membrane depolarization and repolarization. For example, in epileptic foci, which can increase the energy requirements up to fivefolds. This underscores the brain dependence on a continuous supply of oxygen and glucose, as it lacks significant energy reserves. And in case of CO poisoning, carbon monoxide binds to hemoglobin more strongly than oxygen and reduces the oxygen delivery to tissues. This leads to to cause the confusion, headache, dizziness, and sometimes it may more severe cases, coma and death may happen. Similarly, in cyanide poisoning, the cyanide blocks cytochrome C oxidase, a key enzyme in mitochondrial electron transport chain. This stops aerobic respiration, meaning cells including neurons and heart cells which cannot produce ATP. So this leads to rapid loss of consciousness, seizures, and cardiac arrest. And in another example, where the 3-nitropropionic acid, uh, a mycotoxin that irreversibly inhibit succinate dehydrogenase, which is again leads to ATP depletion and motor disorder. So the direct injection of this 3NP in animal models shown neuron degeneration via excitotoxicity. And the neurons are extremely sensitive to energy disruptions due to their high metabolic demands and lack of energy stores. So this makes them a primary target during systemic hypoxia or toxic exposure. The next is axonal transport. Axonal transport is the neuron's internal delivery system. Without it, the brain cells would be like cities with no roads, no couriers, and no way to survive. So let's understand more on axonal transport. So the axonal transport is a cellular process responsible for the movement of organelles, proteins, lipids, and other essential cargoes between the neuronal cell body and the axon terminals. So the transport is the bidirectional transport system is a crucial for neuronal survival, function, and communication, as the neurons rely on it to maintain their structural integrity, delivery signals, molecules, and remove the waste products. So the axonal transport is primarily mediated by two families of motor proteins, that is kinesin and dyneins. The the kinesins are responsible for anterograde transport, which moves the cargoes from the cell body towards the axonal terminals. And the dyneins are responsible for the retrograde transport. That means like the moving cargoes from the axon terminals to back to the cell body. So the proteins and other materials are synthesized in the cell body, especially in the nasal substance, and must be transported to the axonal terminals. So this is where the microtubules and the tau proteins come in. So they provide both structural support and serve as tracks for transport. So axonal transport can be further classified into two main types based on the speed of transport. That one is like fast axonal transport and the slow axonal transport. The first one is this type of transport is responsible for moving membrane-bounded organelles such as vesicles and mitochondria at the rate of up to several hundred millimeters per day. Fast axonal transport is essential for delivering neurotransmitters, receptors, and other signaling molecules to the axon terminal. And whereas the slow axonal transport, this type is responsible for moving skeletal proteins such as tubulin and neurofilaments at the rate of only a few millimeters per day. The slow axonal transport is important for maintaining the structure integrity of the axon and for axon growth and regeneration. So the functions of axon transport is essential. This is essential for delivering materials to the distal axon and synapses, and it also enables retrograde signals, which provides feedback to the cell body. When this system is disrupted, it can lead to neurodegeneration, and as seen in the mutations affecting the dynein and dynetic complex. And also the neuron transport removes the waste products from the axon terminals. This prevents the accumulation of toxic substance that can damage the neurons. And the transport also maintains the structural integrity of the axon. This ensures that the axon can properly transmit signals and support neuronal functions. So the injuries to axon can result in Wallerian degeneration, which we are going to understand in the next coming slides. The process where the distal part of the axon deteriorates. So, moreover, the defects in anterograde retrograde transport are linked to several neurodegeneration diseases, highlighting that the importance of maintaining health axonal transport. Axonal degeneration. Axonal degeneration is a fundamental process in neurobiology, playing a critical role in both injury and disease. But what exactly happens when an axon degenerates? Let's explore the science behind this process. When the injury, as a nerve injury takes place, the nerve cut into two fragments from the site of injury. So the nerve, which is divided into two parts, the distal segment of the injury and the other one is the proximal segment of the injury. So the changes which occur at the distal to the injury was described by the British physician uh, Augustus Weller in 1950. So he described the sequence of events that occur in distal segment of the nerve after it cuts. So the process was named Wallerian degeneration in his honor. So following the axotomy, the distal portion of the axon begins to degenerate. So initially it maintains normal function for a shorter period. This survival time is proportional to the length of the remaining stem. But eventually, the axon undergoes active proteolysis, breakdown its membrane disintegration of the cytoskeleton and mitochondrial swelling, and axonal fragmentation takes place. So this is not a passive decay, but a regulated active process. And and most importantly, the discovery of wild as mouse changed everything. So the wild S mouse is nothing but Wallerian degeneration slow uh, mutant, which was discovered by accident in 1980s in a breeding facility of England. So this wild S mouse is a genetically mutated strain of mouse that shows a dramatically delayed onset of Wallerian degeneration. So in this mouse, injured axons can remain structurally intact and electrically active for up to 2 weeks. So the degeneration is triggered by increased intracellular calcium and activating proteases like calpain 2. These enzymes digest the axon from within and often in an all or none fashion. So in the peripheral nervous system, the Schwann cells respond by differentiating and forming structures called bonds and bugners. so which guide regeneration of axons, and the macrophages, both resident and recruited, clears the myelin debris and secretes the growth promoting factors for the regeneration of axons. So unlike the PNS, the CNS, the central nervous system, lacks this regeneration environment. So in the CNS, the oligonucleotides release the inhibitory molecules, and the astrocytes further impedes regrowth. So this leads to really regeneration chances. And understanding the axonal degenerations opens the doors to therapeutic interventions by targeting the molecular pathways involved. We may one day slow, or one day we may make slow or even reverse the neurodegeneration diseases. Neurotransmission. Neurotransmission is the process by which the nerves communicate with each other or with other cells, like muscle or gland cells. So, in the nervous system, neurons communicate with each other through a specialized junctions called synapses. So, when a electric impulse reaches the end of the neuron, it triggers the release of chemical messengers known as neurotransmitters. So these neurotransmitters process the synaptic cleft and bind to the receptor on the postsynaptic neuron. So this binding can either open ion channel or activate the second messenger system. So ultimately altering the activity of the receiving cells. A classic example of neurotransmission in action is neuromuscular transmission. So here the neurotransmitters acetylcholine is released from a motor nerve and binds to the receptor on a muscle cell. So this interactions leads to the muscle contraction. So it's a highly efficient and rapid process that allows for a precise control of the movement. And the neurotransmissions can be disrupted by chemicals even without visible structural damage of the neuron. So this is an important context of the neurotoxicity. Such disruption can alter the conduction of nerve impulses and affect behavior, cognitive performance, and motor functions. So these chemicals may block transmitter release, interferes with the receptor bindings, or inhibits reuptake or disrupt intracellular signaling pathways. So interestingly, not all alterations in neurotransmissions are harmful. In neuropharmacology, drugs are designed to modulate neurotransmissions to treat conditions like depression, anxiety, Parkinson's disease. For example, sympathomimetic drugs, which mimic the action of the sympathetic nervous system. However, when exposure is excess or inappropriate, these same mechanisms can become neurotoxic. So the therapeutic index helps us understanding the margin between beneficial and harmful effects. Yeah, the next topic is development of the nervous system. So as you know, the nervous system is one of the most complex and vital system in the human body. Its development begins early in gestation and continues well into adolescence. So the nervous system begins forming very early during the first month of gestation in humans, and the process begins with the formation of the neural tubes and foundation of the brain and spinal cord. So this development is continuous through the adolescence and involves a tightly regulated sequence of events, namely cell proliferation, migration to target locations, differentiation into the specific neuron and glial cell types, and formation of synapses, programmed cell death or apoptosis, and finally the myelin sheath formation. So each step is essential for building a functional nervous system. So these processes does not happen uniformly across the brain. The development generally proceeds from back to the front, that is caudal to rostral, uh, with exceptions like cerebellum, where which develops later than the other brain structures develop. So neuroaffective factors, neurotransmitters, and adhesion molecules act as macrophagic signaling, guiding cells to their correct position and helping to form the neuronal circuits. So the developing brain is specifically vulnerable to toxins and toxicants. So this is due to the several factors, the presence of critical windows during which specific structures are formed, and an immature blood-brain barrier, and the underdevelopment endotoxin detoxification systems. So damage during these windows can lead to a permanent structural or functional defects. So, despite its vulnerability, the developing nervous system also has a remarkable capacity of recovery, which is known as plasticity. For example, for the younger animals, often recovery faster from certain neurotoxic insults due to rapid cell turnover and enzyme regeneration. So, however, some effects may remain hidden until later in life when the system is challenged. So, to evaluate developmental neurotoxicity, scientists uses a range of tools like behavioral testings, biochemical assays, and anatomical measurements. Uh, Timing of exposure is critical. Early gestation exposure may affect the brain structure, while the latter exposure might alter neurotransmitter systems or behavior. So, understanding these patterns helps us better assess the risk and protect developing brains. Functional manifestation of neurotoxicity. The functional manifestation of neurotoxicity refers to the observable effects on motor skills, sensory perceptions, autonomic regulation, and cognitive abilities. So these are the final outputs of the nervous system and the most telling signs of the damage. So Tilson, a prominent neurotoxicologist from US EPA, has proposed a two-tiered approach to the functional testing. The tier 1 uses a broad observation tools like motor activity test and the functional observation batteries FOB to detect the neurotoxicity. And the tier 2 involves a refined and targeted test to characterize specific effects. So the behavioral method including event screening and motor activity tracking and the clinical observations, and these tests allows researchers to monitor the onset, progression and reversibility of neurotoxic effects in individual animals over time. And the assessments like electrophysiological tests measures nervous condition, nerve conduction and neurotransmitter integrity. And sensory assessment like the acoustic startle response help pinpoint disruptions in specific neuronal pathways. And the assessments like autonomic functions include cardiovascular assessment and neurotransmitter balance. And the cognitive test, especially important in developmental studies, uses mazes and conditioned responses and operant tasks to evaluate learning and memories. And the lead exposure in children that can lower IQ and the impaired behavior. So similar effects are seen in lab animals, validated these models for the human risk assessment. Functional tests guide researchers to the underlying cellular and molecular mechanisms, and they bridge the gaps between the observable symptoms and the biochemical pathways. Now let's discuss about mechanism of neurotoxicity, which is very much important to understand how neurotoxic compounds affect the nervous system. So in the nervous system, there are four main targets that are commonly affected by neurotoxic agents. Those are neuron cell bodies, axon, myelinating cells, and neurotransmitter system. So each of these target, when damaged, leads to a distinct type of neurotoxicity. So when the neuron cell bodies get damaged, that leads to neuronopathies, which is often irreversible due to the complete loss of entire neuron. You can see here in the picture, you can see the normal neuron cells, which is intact, and in the condition of neuronopathies, the cell body, which is having organelles and nucleus and genetic material, got damaged. Due to that, the entire cell was lost. And in the condition of neuronopathies, where the axon get damaged, and in this condition, like some cases, the axon, which may allows the regeneration, in some cases where the cell body remains intact. And the next one is myelinopathies. The myelinopathies where the myelinating cells will get affected. Those are oligodendrocytes in CNS, or the Schwann cells in peripheral nervous system, and this impairs the signal conduction. And the last one is neurotransmitter systems. The disruption of this causes neurotransmitter associated toxicity, which alters synaptic transmission and the neural communication. So you can see in the picture where the synaptic got disrupted and the communications will majorly affected. So now let's look into the individual neurotoxicity. Neuronopathies. So as we discussed in the previous slide, neuronopathies are disorders characterized by degeneration or dysfunction of neuron cell bodies. So unlike other cells, neurons cannot regenerate once it got destroyed, right? So these neuronopathies can majorly occur in sensory ganglia, anterior horn cells in the spinal cord, and autonomic ganglia. So in sensory ganglia, for example, like when you touch a hot stove, so thermoreceptors and the pain receptors in your skin detect the high temperature and potential tissue damage, right? So these receptors send electric signals through the sensory neurons. Those cell bodies are located in the dorsal root ganglia, that is sensory ganglia. So which are the clusters of the nerve cells, cell bodies just outside the spinal cord. So the neurotoxicants affects majorly these areas, and then that causes sometimes not like non-length dependent sensory loss and sensory ataxia. And the next one is anterior horn cells in the spinal cord. Those are also called as motor neurons, motor ganglia. So these motor neurons are responsible for sending signals from the spinal cord to skeletal muscle. So when the neurotoxicants affect these, it causes progressive weakness, sometimes fasciculation, that is involuntary visible twitching of muscle fibers, and sometimes like it also causes muscle wasting, also known as muscle atrophy. And the neuronopathies also occurs in autonomic ganglia, that is also called as autonomic neuronopathies, which are these autonomic ganglia are located outside the CNS. So they are the part of peripheral nervous system actually. So they serve as relay stations between the central nervous system and the target organs. When these autonomic ganglia got affected, it leads to gastrointestinal dysmobilities, motilities and bladder dysfunction. Sometimes like abnormal sweating may happen, and these neurons are uniquely vulnerable due to the high metabolic rate and long cellular processes and excitable membranes. So these features, while essential for function, but that makes them primary target for cellular toxicants. So many neurotoxic compounds act directly at the neuron cell bodies, right? So let's see some of the examples I have quoted from the book here. So first is doxorubicin. So you know, right? Doxorubicin is a potent chemotherapy agent, and is known for its effectiveness against the cancer, but it also carries a hidden danger, that is neurotoxicity. So in the peripheral nervous system, doxorubicin selectively injures the neurons in the dorsal root and also autonomic ganglia. So the reason behind it, these ganglia cells lacks a protective blood tissue barrier. So this absence allows doxorubicins to penetrate and damage these neurons more easily than the central nervous system. So the doxorubicin first interacts with the DNA transcription and inhibits the topomerase 2 and generates the ROS, reactive oxygen species, and which contribute to its neurotoxic effect. So in the blood-brain barrier, if the blood-brain barrier is breached, then its toxicity spreads to the brain cortex and the subcortex nuclei as well. Yeah, the next example is methyl mercury. So you all already heard about methyl mercury, I hope. See, there are global tragedies with the methyl mercury. If you see, the methyl mercury has caused devastating poisoning in Japan and Iraq. In the mid 20th century, the residents of Minamata Bay in Japan were unknowingly exposed to massive amount of methyl mercury. So industry effluent rich in mercury was rerouted into this bay actually. So the mercury contaminated fish were consumed by the residents, and a widespread of neurological damage has happened. So in a similar way, in Iraq, where the grain treated with methyl mercury as a pesticide, which led over 400 deaths and thousands of hospitalization. So the symptoms of methyl mercury poisoning vary with the age and exposure levels. In adults, majorly the visual cortex and cerebellar granular neurons are mostly affected. So which leads to blindness and severe ataxia. But whereas in children, the damage is more like serious, which includes the developmental disabilities, cognitive deficits, and the mental retardation. So the methyl mercury, which interferes with the multiple cellular functions like glycolysis, nucleic acid synthesis, aerobic respiration, protein synthesis, and even neurotransmitter release. So it also induces oxidative stress and disrupt calcium homeostasis, which contributes to widespread of neuronal injury. Yeah. The next is trimethyltin. Trimethyltin is not commonly encountered in our daily life, but it's an industrial organotin compound that causes selective apoptosis in the hippocampus and cochlear neuron. So in humans, this trimethyltin intoxication has been associated with the limbic cerebellar syndrome, which is conditions marked by memory loss, emotional disturbances, and motor coordination deficits. So similar behaviors as also observed in primates which were exposed with exposed to this TMT, that is trimethylene. So the mechanism behind it is like it induces energy deprivation, excitotoxicity, and upregulates this tumor necrosis factor alpha, which in turn increases expressions of the stannin gene, protein involves in mitochondrial regulation and apoptosis. So see, you can observe here like even which even a structural small molecule which got altered can trigger a complex neurodegenerative pathways. Yeah, now let's try to understand the axonopathies. Axonopathies are a diverse group of neurological disorders that shares a common feature, that is damage or dysfunction of axons. So axons are critical components of the nervous system, responsible for transmitting signals between neurons and from neurons to the target tissues such as muscles and glands. So damage to the axon can disrupt these signals, which leads to a wide range of neurological symptoms depending on the specific nerves affected and the extent of the damage. So this axon like degenerates, the patient develop peripheral neuropathy. So, the symptoms which begin in the hands and feet, following a pattern known as glove and stocking distribution it is. So over time, the damage progresses to more proximal regions and even to the spinal cord. So the long axons, such as those in the posterior columns of the spinal cord, are the peripheral nerves, are the more susceptible toxic damages which causes. So their length increases in the number of potential targets of disruption. So fortunately, when axonopathies affected peripheral neurons, the potential for regeneration is high. But whereas in the central nervous system, the regeneration will be restricted by oligodendrocytes. Yeah. Now let's see the compounds associated with axonopathies. So due to the time constraint, I'm going to touch only the first five compounds, and remaining, however, it is provided here. That is the reason I have given a brief tables to easily understand. So this can be referred anytime if you want. So yeah, let's see the first one. So hexane and gamma diketones. So these hexane and gamma diketones, these like the hexane is a commonly used solvent in the industrial settings. The chronic exposure of hexane, especially in occupational environment, which leads to a progressive sensory motor distillate axonopathy, and sometimes like axonal swelling and atrophy, especially near the nodes of Ranvier. And this often presenting a glow and stalking distribution, which is known as like sensory loss and abnormal sensation like numbness and burning sensations that occurs in the hands and feet. So the key mechanism involves here is like hexane and gamma diketones like metabolize to 2,5 hexane dione. So that further forms a pyrrole adduct, which cross-link with neurofilaments and disrupting the axonal transport. So these long peripheral axons are particularly vulnerable to this hexane and gamma diketones, and this condition is often reversible if exposure is halted early. Yeah, the next one is carbon disulfate. So the carbon disulfate is again another industrial chemical with a similar neurotoxicity profile. So it causes distal axonopathy and can also affect mood and cognition due to the CNS involvement here. So unlike hexane, this CS2, that is carbon disulfate, is directly reactive and forms thiourea crosslinks in the neuron filaments and also erythrocyte proteins. So these crosslinks serve as a potential biomarker of exposure. So that is what like which makes the this carbon disulfate as a well studied model for protein cross-linking in neurotoxicity. And the next compound is acrylamide. Acrylamide is widely used in industry and food processing, and it causes a distal axonopathy and nerve terminal degeneration, and which with the early signs including sensory deficiency and motor weakness. So the major mechanism involved here is like inhibition of axonal transport and accumulation of neuron filaments. Notably, now terminal degeneration often precedes axonal damage, suggesting a primary synaptic lesion. And the last one is organophosphate compounds. So the organophosphate compounds such as triarthropresyl phosphate are no delayed neurotoxicity. Often this condition is called as organophosphate-induced delayed neurotoxicity, that is OPDN. These compounds generally inhibit this neuropathy target esterases, that is NTE, and the toxicity only occurs if the enzymes undergoes aging after inhibition. So the result is degeneration of both peripheral nerve and the spinal cord tract. So unlike cholinergic toxicity, this organophosphate-induced delayed neurotoxicity is non-cholinergic and often irreversible. Okay, now let's discuss about myelinopathies. In the nervous system, myelin acts as an electrical insulator, which enables rapid and precise transmission of nerve impulses. So, but when this insulator is compromised, which results into the group of disorders, which is known as myelinopathies. So this myelin surrounds neuronal process, which allowing signals to travel quickly and efficiently. So when without this myelin, the neurons communicate with ephaptic transmission. This ephaptic transmission means like it forms a communication between adjacent neuron fibers that does not involve any synapses or neurotransmitters. For example, this the cardiac Purkinje fibers, where this ephaptic coupling may plays a crucial role in the conduction of electrical signals in the heart. These myelopathies can manifest in two major forms. One is intramyelinic edema and demyelination. So intramyelinic edema, which is like myelin layers get separated due to the fluid accumulation, and this demyelination, which selectively loss the myelins from the axon. And often this intramyelinic edema may be reversible in the early stage, but if the damage is progressive, then it can lead to the irreversible demyelination. If you see in the central nervous system, this remyelination is limited. We have discussed this previously also. But whereas in the peripheral nervous system, the Schwann cells can regenerate the myelin. Interestingly, remyelination in the PNS involves multiple Schwann cells, which results in a shorter intranodal lengths, which is a permanent marker of the past injury. Let's see compounds associated with injury of myelin. The first one is hexachlorophene, which is commonly used in baby baths. However, it was later discovered to be neurotoxic, particularly in the premature infants. So it was used to prevent the Staphylococcus 6 skin infection, and when it is applied on the skin, it entered into the nervous system, which causes intramyelinic edema and splitting the myelins into layers. And this led to a vacuole formation and a spongy appearance of the brain. So which was also disrupted ion gradient and mitochondrial function, further contributing to myelin damage. When this hydrochlorine was taken high doses, it can lead to axonal degeneration and damage to retina photoreceptors, and which will further affect the vision. So functionally, this toxicity shows up as a generalized weakness and confusion and seizures. If the conditions progresses, then it leads to coma or even death. The next one is tellurium. Tellurium is generally used as a in metallurgy and explosives, and tellurium is added to copper and stainless steel to improve machinability. There are no well documented cases of human exposure of tellurium, but annual studies which revealed that tellurium can cause the neurotoxicity, particularly in the peripheral nervous system. So in the young rats, it disrupts myelin lipid synthesis in Schwann cells, particularly affecting the cholesterol and cerebrocytes. So tellurium interferes with the conversions of sequelin to cholesterol, which likely be by inhibiting sequelin epoxidase, a key enzyme in this pathway, and then this leads to a lipid accumulation and Schwann cell dysfunction. The last one is lead. So lead is a heavy metal whose neurotoxicity has been recognized from the past like centuries. So today, like the lead exposure still occurs in occupational setting, from plumbing and paintings to like fuels and alloys. So the children's absorb the lead more efficiently because of their lack the fully developed blood-brain barrier, which make their brains more susceptible. And in animal studies, the lead exposure results in peripheral neuropathy, marked by segmental demyelination, a process that closely resembles the effects of another toxic metal, that is tellurium. So when the chronic exposure of this lead in adults, which develops peripheral neuropathy along with the symptoms like abdominal pain, anemia, and lead depositions in the gums and bones. Yeah, astrocytes. Astrocytes are a type of ganglion cells, which we have already discussed in the previous slides, where we covered the different cells of supporting cells of central nervous system. So it is one of the central nervous system glial cell, which is named for their star-like shape, and they play a crucial role in supporting the brain function and responding to the injuries. So astrocytes regulate neurotransmitter levels, buffer ions, and maintain a blood-brain barrier, and metabolize both endogenous and foreign substances. So their role in spatial buffering and metabolic processing and detoxification makes them frontline defenders in the central nervous system. So astrocytes are absorb excess neurotransmitters like glutamate, which prevent the excitatory toxicity. And here we should know about the glutamate and glutamine cycle, which is a metabolic pathway that occurs between the neurons and astrocytes. So it which ensures the recycling of glutamate, the brain's primary excitatory neurotransmitter that prevents neurotoxicity due to the excess of glutamate. So what will happen in this is like the in the presynaptic nerve, this glutamate is stored in vessels, which is like upon stimulation, it releases into the synaptic cleft. So the glutamate binds to the receptors present on the postsynaptic nerve, and which triggers the excitatory signal. And whatever the excess glutamate present in the synaptic cleft, so that is taken up by these astrocytes via excitatory amino acid transferase, that is EATTS. So this prevents the excitatory toxicity, which can damage or kill the neurons. So this whatever the glutamate taken by the astrocytes again converted into glutamine, and with the help of enzyme glutamine synthase. So this glutamine again, which is the safest and non-toxic glutamine that transported again back to the presynaptic nerve by the astrocytes. So which is again the in the neurons, this glutamine is converted back into the glutamate by again the enzyme glutaminase, and which is again uh, like participate in the glutamate-glutamine cycle. This way, like, it prevents the excitatory toxicity and maintains neurotransmitter balance, and support neuron astrocyte metabolic cooperation. Yeah, the compounds associated with astrocytic effect, uh, ammonia. Ammonia, like when the liver fails, it can no longer effectively remove the ammonia from the bloodstream. So this leads to excessive ammonia accumulation in the brain. So the effects of ammonia can on the central nervous system depends on its concentrations. Like if it is like at higher levels of ammonia, which has a depolarization effect on neurons, which can trigger seizures. Whereas if the lower levels, it causes hyperpolarization on neurons, and which leads to stopper and coma. So one of the earliest and most significant changes in the brain during the ammonia intoxication seen in astrocytes is the cells swell and undergoes morphological changes, forming Alzheimer type 2 astrocytes. So these changes occurs before other structural damage in the brain. So these cells detoxify the glutamate into glutamine via glutamine synthase that we already discussed in the previous slides. But this excess ammonia disrupt this process, which the glutamate to glutamine process, and impairs the neurotransmission and astrocyte neuron signaling. The next one is nitrochemicals, which are used in medicine and various other industries. This also triggers the astrocyte. Uh, One such compound, that is 1,3-dinitrobenzene, causes gliovascular lesions in the brainstem and cerebellar nuclei. Both the regions are rich in astrocytes. So this dinitrobenzene is bioactive by NADPH dependent cytochrome C reductase, which leads to oxidative stress and mitochondrial dysfunction. This and also this astrocyte is in astrocytes in high glucose demand areas are vulnerable, especially for this nitrochemicals. This may be because of the differences in the mitochondrial respiratory capacity and the antioxidants. Next is metronidazole, which is a common antimicrobial, which can also cause peripheral neuropathy and encephalopathy when it is used long term, at the high doses. And this can cause the symptoms which include seizures and hallucination. So metronidazole, which reduces to a toxic hydroxyamyl intermediate, which binds to the cellular macromolecule. So its structure resembles thiamine, a vital nutrient for mitochondrial metabolism that suggests anti-metabolic metabolite mechanism of neurotoxicity. The next one is methionine sulfoxamine, MSO. This MSO is a potent inhibitor of glutamine synthase. This is a vital enzyme responsible in converting glutamate to glutamine. So this ingestion of large amount of methionine sulfamine leads to a neuronal cell loss, in the hippocampal facial dentata and pyramidal layers, cerebral cortex, and Purkinje cells of the cerebellum. So these astrocytes will begin to swell, and their mitochondria become damaged, and glucogen level spikes, particularly within astrocyte cell bodies when the high concentrations of methionine sulfoxide uh, exposure. So this MSO inhibits same like glutamine synthase, the synthase of glutamate and GABA, the key neurotransmitters involved in excitatory and inhibitory signaling. So this imbalance may contribute to seizure generations. So although the exact mechanism remains unclear till. Yeah, the last one is fluoroacetate, which is found in certain plants and used as a rodenticide. And fluoroacetate and its metabolite fluorocitrate both are potent inhibitors of the Krebs cycle. So these compounds are preferentially taken up by the glial cells, including astrocytes. The fluorocitrate is metabolized to fluoroacetyl-CoA, which condense with oxaloacetate to form the fluorocitrate. And this fluorocitrate inhibits akinetase, leading to citrate accumulation, ATP depletion, and mitochondrial dysfunction. So in animal, like when the animals exposed to this fluoroacetate, which experience the tonic convulsions within minutes. So even after like the survivor animal, they show the respiratory arrest and cardiac failure. So this astrocyte shows disrupted glutamate synthase. It is not due to the ATP depletion, but due to the rerouting of 2-oxoglutarate into the TCA cycle. Neurotransmission associated neurotoxicity, which refers to the harmful effects of the certain substances exert on the process involved in the transmission of signals between the neurons. So these processes include neurotransmission synthesis and release, receptor interactions, and reuptake. So the pesticides, organophosphate and carbamide, which are widely used insecticides, which poses significant risk on human health. So both these compounds exert their toxic effects on primarily by inhibiting acetylcholinesterase enzyme, which is responsible for breaking down the acetylcholine into choline in the synaptic cleft. So when this acetylcholinesterase is inhibited, the acetylcholine which accumulates and lead to cholinergic overstimulation. So this overstimulation can manifest the range of symptoms, starting with mild flu-like signs, and when it gets progressive, more severe outcomes such as gastrointestinal distress, muscle twitching, ataxia, convulsions, coma, and in extreme conditions, death. So interestingly, the severity of symptoms does not always correlate directly with the degree of cholinesterase inhibition. So this has led researchers to uncover additional mechanism of toxicity. That additional mechanism involves the altered reuptake of choline, which is a precursor for acetylcholine synthesis. So disruption in this process can further like impair the imbalance in cholinergic signaling. So here are some of the compounds produce neurotransmission associated neurotoxicity. Yeah, nicotine, which is widely available compound found in tobacco products and certain pesticides. So while it's best known for its addictive properties, nicotine also has a wide range of pharmaceuticals and toxic effects. So nicotine primarily acts by its binding to nicotinic acetylcholine receptor, which are found in autonomic ganglia at the neuromuscular junctions, and within the central nervous system. So this interactions is responsible for both its stimulant and addictive properties. So in the central nervous system, the nicotine alerts electroencephalography graphic activity, and these changes can be blocked by mecamylamine, which is a nicotine receptor antagonist, uh, and this nicotine affects largely due to the stimulation of the sympathetic nervous system, and which leads to the increased heart rate and blood pressure, uh, vasoconstriction in skin, and a paradoxical sensation of relaxation. And the nicotine, these paradoxical responses can be blocked by alpha and beta adrenergic antagonist, confirming the role of sympathetic activation. So this nicotine toxicity contributes to cardiovascular diseases, mainly increasing heart rate, blood pressure, and platelet aggregation, and cancer risk, um, and possibly by inhibiting apoptosis and promoting tumor growth, and neurodevelopmental issues in children born to the mothers who smokes during the pregnancy. The one is cocaine and amphetamine. So beyond nicotine, other stimulants like cocaine and amphetamines pose a significant health risk despite their illegal status. Usage remains widespread. So the mechanism behind both these chemicals compounds are like cocaine primarily acts by blocking the reuptake of dopamine, norepinephrine, and serotonin to the nerve terminals in the central nervous system. This lead to a accumulation of these neurotransmitter in the synaptic cleft, particularly the dopamine, which is responsible for drug euphoric and addictive effects. So in contrast, this amphetamine stimulates the release of dopamine and norepinephrine, and can directly damage mono and aminergic neurons. They also affect serotonin system, especially with the drug like MDMA, leading to long-term depletion of serotonin terminals. So this cocaine enhances dopaminergic transmission by inhibiting the dopamine transporters, resulting in increasing self-confidence, euphoria, and in some cases paranoid psychosis. So these effects are closely linked to dopamine D1 receptor activity. So in amphetamine, on the other hand, produces uh, heightened alertness, energy, and emotional worth, but they also carry a risk of neurotoxicity, particularly to dopamine and serotonin axons. So chronic use of amphetamine can lead to behavioral disturbances and cognitive deficits due to the long-term neurotransmission depletion. Chronic use of cocaine is associated with the cerebral atrophy, increased risk of stroke, and vascular abnormalities, whereas the amphetamine are more directly neurotoxic, causing oxidative stress through dopamine oxidation, and which generates free radicals that damage neurons. So while cocaine neurotoxicity is less directly linked to the oxidative stress, amphetamines are strongly associated with the oxidative stress. So both the cocaine and amphetamine have been shown enhance the toxic effects of HIV1 proteins such as TAT and GP120. So these interactions contribute to the development of HIV-associated dementia. The combined toxicity is believed to be mediated by oxidative stress, and interestingly, beta estradiol has been found to reduce this synergic damage. Yeah, the next one is excitotoxins. In the uh, intricate web of the human brain, communications is key. Among the many messengers, glutamate stands out as a peripheral, as a principal excitatory transmitter. It fuels thoughts, memories, and movements. But under certain conditions, it can also become a silent killer. So this glutamate and other amino acids activate excitatory amino acid receptors. Uh, these receptors come in two main types. One is inotropic, which directly opens ion channels, and metabotropic, which works through G protein. Among the inotropic receptors, NMDA, MPA, and kainate receptors plays crucial role. So in controlled experiment, scientists discovered that high concentration of glutamate can be neurotoxic. So this phenomena known as excitotoxicity, which is linked to the conditions like hypoxia, epilepsy, and neurodegenerative diseases. So this when the glutamate injected into the infant rodents, glutamate targets areas where the blood-brain barrier is weakest. So here it opens ion channels, causing neuron to swell and die, affecting dendrites and cell bodies, but it sparing the axons. In humans, the only known mild reactions is so-called Chinese restaurant syndrome. This where the sensitive individuals experience burning sensations after consuming large amount of monosodium glutamate. In another chemical called kainate, found in seaweeds, is even more powerful than glutamate. Scientists use to it study a brain because it can destroy specific brain cells without harming nearby connections. Yeah. Next is toxicant-based models of neurodegenerative diseases. So this particular slide will give overview of toxicant-based models used in the study of neurodegenerative diseases. So these models utilize specific toxins to mimic the pathological hallmarks and observational deficit observed in the conditions like Parkinson diseases, Alzheimer diseases, and Huntington's diseases. So by understanding these toxin-induced neurodegeneration, researchers can gain valuable insights into the disease mechanism and develop potential therapeutic strategies. So yeah, one of the toxicant-based model is MPTP, which is methylphenyl tetrahydropyridine, which was discovered accidentally in the early 1980s when the individuals using a synthetic opioid contaminated with this compound, which developed symptoms nearly identical to Parkinson's disease. So these symptoms includes like rigidity, bradykinase, and in some cases complete immobility. The mechanism behind this MPTP is like it is the compound is lipophilic and which crosses the blood-brain barrier with ease. And once it inside the brain, it is taken up by the astrocytes where it is get metabolized and by the enzyme monoamine oxidase B, and which further forms into methylphenyl dihydropyridine, and this further oxidizes into methylphenylpyridine, which is an active neurotoxic compound. So this MPP plus, that is methylphenylpyridine, is selectively taken up by the dopaminergic neurons in the substantial nigra via the dopamine transporter. So once it is inside, it inhibits the mitochondrial complex 1 which leads to the impairment of ATP production and increased reactive oxygen species. So this results in oxidative stress and ultimately which causes neuronal death. So the while this dopaminergic neurons are the primary targets, noradrenergic neurons in the locus ceruleus are also affected with the repeated exposure. But however, these are less sensitive to single doses. So interestingly, this monoamine oxidase B inhibitor, like selegiline, can protect against the methyl phenyl tetrapyridine toxicity. This protection is not solely due to the enzyme inhibition. It is also involved the antioxidants and free radical scavenger properties. There is a species difference. Like rats are notably resistant to MPTP induced neurotoxicity. So this is due to their endothelial cells converting this MPTP into the polar metabolites like MPTP and oxidase, which do not crosses the blood-brain barrier. So the methylphenyl tetrapyridine has provided a powerful model for studying Parkinson's disease. It has helped us understanding the role of mitochondrial dysfunction, oxidative stress, and selective neuronal vulnerability in neurodegenerations. Yeah. The next model is manganese. So we all know that manganese is an essential trace metal which found in all the tissues of humans and animals. And the manganese plays a crucial role in metabolism of amino acids, proteins, lipids, and carbohydrates. So the manganese functions as a cofactor for several important enzymes, which includes glutamine synthase and also the manganese superoxide dismutase. These enzymes, which helps in protecting the cells from oxidative damage. So in rare cases of deficiency, the manganese produces the symptoms such as seizures, poor growth, skeletal deformity, and even some reproductive issues. In the case of excessive exposure of manganese, it can be very harmful, and particularly to the nervous system. So which the excessive exposure which tends to accumulate the manganese in basal ganglia, affecting dopamine producing neurons and particularly disrupting other neurotransmitter systems. So the common source of this manganese exposure includes the industrial activities, which includes welding, mining, and the steel production, as well as the food contamination and food and water and certain pesticides also causes this manganese toxicity. So the symptoms which include by the manganese toxicity is also known as manganism, that can resemble the Parkinson's disease and include physiological which includes the physiological disturbances, motor dysfunctions like tremors and rigidity, and also the behavioral changes. So unfortunately, the early detection remains a challenge in this case, as the reliable biomarkers are still lacking to detect. So the neurotoxic effects of manganese are linked to the mitochondrial dysfunction, oxidative stress, and protein aggregations. So all of which contribute to the health of dopamine neurons. So the manganese accumulates in the brain regions such as substantia nigra and globus pallidus and striatum. Interferes with the ATP production, similar to the effects of mitochondrial poisons. So these areas are particularly vulnerable due to their high metabolic activity and this manganese transporter distribution. So understanding the balance between manganese essential functions and its potential toxicity is crucial for protecting both public health and workers in high-risk environments. Yeah. The next model is Gone syndrome. It is guanine cyclid-induced Parkinsonism and amyotrophic lateral sclerosis syndrome. So in the early 1900s, an unusual high rate of hereditary paralysis was observed among the natives of Guam. So by the mid of 1950s, the incidences of this amyotrophic lateral sclerosis and unique conditions known as Parkinsonism dementia complex was found to be up to 100 times higher on Guam than anywhere else in the world. So while the Guam ALS closely resembles the ALS seen in globally, and Guam Parkinson's dementia complex is distinct. It combines both like symptoms of Parkinson's disease and dementia. So this appears simultaneously. So the clinically the affected individual showed mental decline, motor neuron damage, and some of the Parkinson's features. And so neuropathological studies also revealed the cortical atrophy, loss of pigmentation in the substantia nigra, and widespread degeneration of ganglion cells throughout the central nervous system. So researchers suspected an environmental cause possibly linked to the consumption of seeds from the cycas plant, commonly known as the cycad. So these seeds contain toxic compounds such as cyclosin and beta methyl aminoalanine. So the cyclosin is metabolized into a carcinogenic and a liver toxic compound, which is known as MAM, that is methyl azo methyl azoyl methanol. While this beta methyl aminoalanine is a non-protein amino acid that acts as a glutamate excitotoxin, which damages the neurons by overstimulating the glutamate receptors. So the BMAA toxicity is similar to glutamate's effects and can be blocked by the specific receptor antagonist. It also increases intracellular calcium levels through a beta carbamate intermediate, which further contributes to excitotoxicity and may trigger the mitochondrial permeable transition pore, which is a key pathway in the neurodegeneration. Environmental factors relevant to neurodegenerative diseases. So in this slide, we explore how environmental exposure contribute to the development of neurodegenerative diseases, particularly Parkinson's disease and Alzheimer's disease. So while genetics play a crucial role, especially in the early onset cases, environmental factors are increasingly recognized as a major contribution to the late onset and sporadic forms. So, in the discovery of methylphenyl tetrapyridine induced Parkinsonism was a turning point, where which led to a surge in research into environmental cases of Parkinson's disease. So the epidemiological studies have linked the Parkinson's disease to exposure to pesticides like paraquat and organophosphates, as well as metals such as manganese and its solvents. So these substances are commonly encountered in agriculture and industrial settings. So many of these toxicants share a common mechanism, which is they impair the mitochondrial functions and increase oxidative stress. So which leads to the death of dopamine producing neurons. So this complex 1 inhibitors like methyl phenylpyridine which are used experimental models to replicate the Parkinson's disease pathology. So interestingly, some lifestyle factors appears to be protective, such as cigarette smoking and caffeine consumption, have been associated with reduced risk of Parkinson's disease. So this may be due to their effect on mitochondrial integrity and mono amino oxide inhibition. And a unique case in the Gone syndrome was observed. Like initially, this was thought that it is caused by genetics, but later it was linked to environmental exposure, especially the consumption of flying foxes that fed on the cycad seeds containing this neurotoxic uh, compounds. And the syndrome shares similar features of the Parkinson's disease and Alzheimer's disease. And although the AD was less studied, but it also associated with the environmental exposure. Aluminium in drinking water and pesticide exposure has been implicated. A notable examples is the dialysis related dementia, and which was linked to the aluminium in dialysate. So overall, the evidence strongly supports a role of environmental exposure in the pathogenesis of neurodegenerative diseases. So understanding these links not only helps in identifying at-risk populations, but also opens avenues for preventing and interventions. Developmentally neurotoxic chemicals. So there are a wide range of substances have been implicated in developmental neurotoxicity. These includes heavy metals like lead and mercury, solvents, pesticides, pharmaceuticals, ionizing radiation, and persistent organic pollutants such as polychlorinated biphenyls and polybrominated diphenyl ethers. So the timing of exposure is crucial in developmental neurotoxicity. Even a chemical that may be relatively harmless to the adults, that can adversely affect on a developing brain. So this is because of the developing nerve system is undergoing a rapid and highly coordinating changes. So let's see some of the examples of developmental neurotoxicants. Ethanol. So prenatal exposure of ethanol leads to a fetal alcohol syndrome characterized by abnormal neuronal migration and dendritic spine malformations and widespread apoptosis. So this results in cognitive deficiencies and delayed myelination. And the next example is lead. We already studied about lead. There is like no safe, there is no known safe levels of lead exposure. Even low levels of lead have been linked to the reduced IQ and behavioral changes. So the lead interferes with the neurotransmission and synaptic development. And the other examples like methyl mercury, which we already discussed, that is which it is a famous incident of a Minamata Bay disaster. I'm not going to touch here. And the nicotine, cocaine also we have discussed. So the persistent pollutants such as polychlorinated biphenyls and polycarbonated diphenyl ethers, uh, which disrupt the endocrine function, particularly the thyroid hormone signaling, which is a vital for the brain development. So the affects the neurotransmitter systems and cognitive function. And the developmental neurotoxicant poses a significant public health concern. Their effects are often irreversible and can have lifelong consequences. So continued research, regulation, and public awareness are essential to protect the vulnerable populations, especially during early development. The next is chemical that induce depression of nervous system function. So the CNS depressants are substances that slow down the brain activity. Interestingly, at low doses, they may first suppress inhibitor signal and leading to apparent excitations like the intoxication seen with the alcohol. But as the dose is increased, the effects progress for sedation and motor impairment to coma. And in extreme conditions like death, the death happens due to the respiratory depression. And the wide range of volatile solvents can cause generalized CNS depression. Those are includes aliphatic and aromatic hydrocarbons, halogenated hydrocarbons, ketones, esters, and alcohols and ethers. So these small and lipophilic molecules are found in every products of daily life. So nail polish removers, paints, industrial solvents, and even anesthetic gases. So the key receptors involved in this is the GABA and NMDA and glycine receptors. So these are the these are the same targets affected by drugs like barbiturates and benzodiazepines. So the long-term exposure to solvents can lead to conditions known as solvent-induced chronic toxic encephalopathy. The symptoms include irritability, fatigue, and memory loss issues. Yeah, the next is in vitro and alternative approach to neurotoxicology. So the traditional neurotoxicological assessments have heavily relied on in vivo animal models. But however, these models present several limitations, which includes ethical concerns regarding animal welfare. The animal studies are high cost and time consuming procedures. And also it is difficult to extrapolate findings to the humans due to the species differences. So consequently, there are a growing need for alternative approaches that can provide reliable and relevant information about neurotoxic potential while like minimizing or replacing the animal use. So among that, the high throughput analysis is one of the alternative approach. In 2007, the National Research Council introduced a transformative vision for toxicity testings in the 21st century. So the goal of this National Research Council was to move away from the traditional animal-based testing uh, towards the high throughput in vitro methods and computational models like QSAR, that is quantitative structure activity relationships. So these methods aim to predict adverse outcomes by screening thousands of chemicals quickly and cost effectively. So to manage the vast number of untested chemicals, a tiered testing strategy has been proposed. So the first tier, which uses the high throughput assays to detect interactions with the key biological receptors. So if the chemical is flagged in this stage, then that can be tested in alternative species like zebrafish or invertebrates, and which offers moderate throughput in an intact nervous system. So finally, only the most concerning chemicals would proceed to mammalian testings, which would be tailored based on the earlier findings, such as the focusing on myelin-related endpoints if earlier data suggested only the demyelination. So this approach is not only like improves efficiency, but also ensures that each method contributes unique insights, helping refine when and how the traditional animal models are used. Another approach is cell culture and model organism approach. So cell culture system are essential tools in neurotoxicology. They allows us to study how toxin affect intracellular processes like signaling, metabolism, and neurotransmitter dynamics. So these systems offer precise control and enabling techniques like patch clamping to study electrophysiological properties but however, they do have limitations such as the loss of complex cell interactions and signaling pathways that are present in vivo. So to address these, researchers are increasingly turning to non-mammalian model organisms like sea algae, zebrafish, and fruit flies. So these models offer the advantage of studying toxicity in all organisms, and where the complex interactions and metabolic processes are preserved. So their embryos are often transparent, so which allows real-time observations on neuronal development. So moreover, these species are genetically traceable, and they have fully sequenced genomes, so which enables the high throughput screening using tools like mRNA interfaces, microarrays, and gene expression profiles. So together, these in vitro and alternative organism approaches provides a powerful integrated framework for identifying and understanding the neurotoxicants. The next is stem cell based approaches. So recent advances in human and mammalian stem cell technology have revolutionized how we study the neurotoxic effects of chemicals. So these technologies now allows researchers to examine toxicity across different stages of brain development. So from early formation to maturity using in vitro models. So a major breakthrough was the discovery of this radial glial cells, which express brain lipid binding protein, which gives rise to most neurons and astrocytes in the developing central nervous system. So these cells persist this postnatally as adult neuron stem cells in regions like the subventricular zones and subgranular zones, making them valuable for modeling neurodevelopmental process and testing the effects of toxicants. Neural progenitor cells, which are derived from both animals and humans, have been used to study the chemicals affect neurons, astrocytes, and oligodendrocytes. This three-dimensional neurosphere culture have future enhanced our ability to model complex developmental events such as cell proliferation, migration, and differentiation. So these models have revealed species differences in vulnerability to toxicants. So, for example, the flame retardants like polybrominated diphenyl ethers affect oligodendrocytes development differently in mouse versus humans. So, such findings underscores the importance of using human relevant models in neurotoxicity research. So the major leap forward come with the development of induced pluripotent stem cells, that is iPSC. These are created by reprogramming adult cells such as fibroblast into stem cells capable of differentiating into any cell types, so including neurons. So these cells retain the donor's clinical and exposure history, so making them ideal for the personalized neurotoxicity studies. So these models not only accelerate the identification of neurotoxicants, but also reduces reliance on animal models, offering a powerful tool for public health research. The next is systems and computational approaches. While in vitro and stem cell models are powerful, system and computational approaches are essential for scaling our understanding from molecule to whole organisms. So advances in imaging, sensing, and computational powers are enabling us to stimulate and analyze complex neurotoxicological systems. So these models incorporate key toxicological principles such as dose responses, pharmacokinetics, and also the metabolism. So computations is also are already improving risk assessment and hazard prediction. And as a technology evolve, they will become even more integral to neurotoxicological research. The last topic is epigenetics in neurotoxicology. Epigenetics refers to heritable changes in gene expressions that occur without altering the DNA sequence itself. So these changes such as DNA methylation and histone modification and regulation by non-coding RNAs, which plays a crucial role in controlling when and where the genes are expressed. So in the recent years, toxicologists have begun to explore how environmental exposures, especially during the early development, can lead to the long-term health effects through the epigenetics mechanism. So this concept known as fetal basis of adult diseases, which suggests the early exposure to toxicants, which may uh, predispose individual to neurological disorders later in the life. So, for example, children born during the Dutch famine showed increasing risk of schizophrenia and decreased methylation of the IGF2 gene. So, which highlights how prenatal conditions can influence lifelong brain health. So, one of the earliest links between epigenetics and neurodevelopmental disorders was the discovery that Rat syndrome is caused by mutations in the methyl CpG binding protein, so which regulates the gene expression through DNA methylation and histone interactions. Which since then researchers has shown that epigenetics changes can contribute to a wide range of neurological conditions, which includes schizophrenia, Alzheimer's disease, and Parkinson's disease. So the environmental toxins such as heavy metals, endocrine disruptors, and pesticides have been shown to alert the epigenetic markers. For instance, lead and mercury exposure during during development can change the DNA methylation and histone acetylation pattern, which leads to the behavior and cognitive deficits. In similarly, the exposure of substances like bisphenol A and methoxychlore has been linked to epigenetics changes that affects the reproduction and neurological health. And also substances like alcohol, cocaine, nicotine, and methamphetamine has been shown to alert the DNA methylation and histone modifications in brain regions, which associated with addiction and behavior. So these changes can influence gene expression and neural function, sometimes in way that persist long after exposure. For example, cocaine has been shown to increase expression of genes like CFOS and the BDNF through the histone acetylation, and blocking these epigenetic changes can reduce the drug-seeking behavior. So, although researchers in this area is still evolving, but it's clear that epigenetics mechanisms are powerful lens through which we can understand how environmental exposures shape brain development and functions across the lifespan. Thank you all for your time and attention. I truly appreciate your interest and engagement throughout this session. Yeah, thank you once again.